Hi everyone, welcome to
amazing channel of Brilliant Katha.
When you're rubbing one body with another body, electrons are transferred from one object to another object. Okay, this process, this type of electricity is been given a name.
halogen connected carbon sp3, then it's connected to benzene. See all of you, halogen connected. What is this carbon hybridization? Full single bond sp3, see then a benzene ring.
Denominator poles shouldn't be included in the denominator poles. That means the denominator, these -1 and -5 should never be included in the solution.
Homocircle caudal fin. It's giving two equal lobes. But in case of cartilage fishes, look at here, one big lobe is there, one small lobe is there. You cannot cut into two equal half this fin.
Hi everyone, welcome to the classes of Brilliant Katha. Today we are going to discuss two important chapters from biology, that is biotechnology as well as microbes in human welfare. The most important area we will be going through, and we will try to revise that topic also. So let's start with the discussion. Sandeep sir.
Yes, hello my dear students, good evening you all. So far we have been discussed different type of questions, right? Different type of chapters also. Principles we completed, molecular basis we completed, and yesterday we completed what? Human health and disease, sexual reproduction, flowering plant. In this class, today's class, two most important chapter. One is called biotechnology principle and process, and second one is called microbes in human welfare.
Hello students.
Two chapters.
Please comment whether it is audible.
Yes.
We just want to see. Please comment.
Am I audible, students? Am I audible?
Are we audible? Please make a comment.
Yes. Okay.
Yes.
Right.
So we'll be starting with the first chapter, that is biotechnology principles and process. So let's start with the question discussion.
Right.
Sir.
Students, look at here. The first question from my biotechnology principle and process. Before I go to this question, my dear students, everyone keep in mind, this is the most important chapter. Biotechnology twin chapter occupy what? 12 marks in CBSE examination, and also in NEET purpose also most important questions are coming from this chapter. So what are the important topics you should focus on in this chapter? Especially restriction endonuclease enzymes. Number 2, RDNA technology, all steps. Number 3, PCR also, PBR 322 also. These all are most important concepts. Everyone thoroughly prepare before NEET exam and before final exam of CBSE. Look at the first question. The question tells us, which of the following steps are catalyzed by Taq polymerase in PCR reaction? The given PCR, Taq polymerase enzyme, Thermus aquaticus from hot spring bacteria. What is that? Archaebacteria. From that bacteria we isolated one complicated enzyme. The enzyme called what? Taq polymerase enzyme. This Taq polymerase enzyme in PCR reaction they are asking denaturation of template DNA, aligning of primers of template DNA, extension of primers and on the template DNA, and reverse transcription in virus is given. Can any one of you give? Very good, absolutely correct. Yes. Who is that? Who is that? Sir, first, they are absolutely correct. We know in PCR 3 steps are there. What are they? One is called denaturation, second one is called annealing, third one is called extension. In these 3 steps, extension process is that process when you add the Taq polymerase enzyme. Actually, what is Taq polymerase enzyme? DNA polymerase enzyme. DNA polymerase enzyme have an ability to do what? Copy, polymerization of new DNA. So our answer is what? Option C, extension process, 72 degrees Celsius of temperature. So exactly at this temperature we need a thermostable enzyme. So where we are getting from? That enzyme is coming from Thermus aquaticus bacteria. What is the enzyme name? Taq polymerase enzyme. So first question answer is option C is the first question answer. Look at the next question.
Now, next question will be from microbes in human welfare. So you might have learned the structure of the bacteriophage. You know what is a bacteriophage? The virus that is attacking a bacterial cell, that is actually called as bacteriophage. And if you take the structure of a bacteriophage, you should understand this portion only will be having the genetic material. What is that region? That is actually the head region. Then second, you can see there will be the second region that will be coming here, B, that is actually the collar. Then the C that has been given is the tail, and you can see D is the end plate. And in that you can see the end plate will be having tail fibers, which include the small one, that is pins. The small fibers are actually called pins, and the long one is actually called as prongs. So this is the structure of a bacteriophage, and hope you know the life cycle of a bacteriophage also. In this virus you can see whenever they are attacking the bacterial cell, so bacterial cell will be looking like this. Whenever this bacteriophage is attacking this, they will be injecting the genetic material into this bacterial cell. So after injecting the genetic material, you can see they will go and fuse with the bacterial genome. 2 things can happen. Either when this bacterial cell is multiplying, you can see they will remain silent, and along with that the viral genome also will multiply. This phase of the life cycle is called lysogenic phase. Or else second thing that can happen is they will convert the cell into a virus factory, in which they will generate a lot of virus inside the cell, and they will slowly lyse the cell or burst the cell. So this is actually called, by bursting the cell, they will be released out. This phase of the life cycle is called lytic phase. So this much you should be knowing about the bacteriophages. So you know what is the answer. Here head, collar, tail and the end plates, that is coming in option 3. Hope you got it. Many of them has commented. Now let's move to the next question.
Good students, keep messaging, but if you know answer, you just try to message in the inbox. While isolating DNA from bacteria, which of the following enzyme is not required? The mention. Which of the following enzyme is not required? You know, in the last class we discussed, remember all of you, if you are isolating DNA from bacteria, if you are isolating DNA from plant cell, if you are isolating DNA from animal cell and also fungi cell, we will use different type of enzymes. You got my point? Different type of enzyme. To lytic, that means to isolate DNA from bacteria, which enzyme we will use? Tell me. Lysozyme enzyme we will use. And from the plant cell, cellulase enzyme. Very important. Animal cell is what? Protease enzyme. Fungi cell is what? Chitinase enzyme we will use. But they are asking which of the following enzyme is not required the mention. Not required the mention. Now here it is: lysozyme enzyme, ribonuclease enzyme, deoxyribonuclease enzyme, protease enzyme. If you use deoxyribonuclease enzyme, DNA will be die. DNA will be completely destroyed. You got my point? So if you use the lysozyme enzyme, okay, that entire the outer border will be get out, that will dissolve. Ribonuclease enzyme, RNA will be destroy, no problem, and protease also no problem. But if you use deoxyribonuclease enzyme, what happened? That DNA will be degenerate. You are looking for DNA, right? You're looking for the DNA from bacterial cell. You wanted to take out the DNA from bacterial cells. So what would happen if you use the deoxyribonuclease enzyme? That enzyme will be destroy or dissolve the DNA. So which is not required, my dear student? Tell me. Absolutely correct. Deoxyribonuclease enzyme is not required. Sabas, very good, very good. Next question.
Coming to the next question, what is the beneficial role of lab in our stomach? So if you are taking the beneficial role of lactic acid bacteria or lactobacillus, you can see they will cause souring of milk by decreasing nutritional quality is actually wrong. They actually increase the nutritional quality. They are not decreasing the nutritional quality. Already so many are answering. Now second one, they increase the amount of vitamin D is actually wrong. It's actually vitamin B12 they are actually increasing. Then third, it checks the disease causing microbe. That is actually the beneficial role. Then it produces alkali which coagulates. It's not alkali they are producing, it's producing acid. So understand the role of lactobacillus. So lactobacillus that is added in a milk will be converting the milk sugar lactose will get digested into galactose and glucose. You know they are joined to each other by 1,4-glycosidic bond. Later it will be getting converted into what? It will get converted into lactic acid. This lactic acid will be performing coagulation. They will be clotting the milk protein, that is casein. So there is coagulation of casein and it will be getting clotted, and because of that only you can see the milk is getting converted into curd. So this is curd formation by lactobacillus. So which vitamin is actually increasing? Vitamin B12. Hope you got it. Many comments actually came. Okay, good one. Now let's start with the next one. Sir.
Next question. Look at the next question. Which of the following should be chosen for best yield if one were to prove or produce a recombinant protein in large quantities or large amount? Which one of this one you're using to produce the recombinant protein in large amount? It's given. One is called laboratory flask or largest capacity, a styrate tank bioreactor without inlets and outlets is given. Without inlets and outlets. If it is without inlets and outlet, how can it work, my dear students? Right? Next one is called what? A continuous culture system, and all of the above is given. What is this culture system? You know, culture medium we discussed in the theory class. Culture medium consists the essential substance for the growth of bacteria. We know this one is what? Tell me. Recombinant protein, nothing but what? Where is the recombinant protein? Wherever the bacteria, inside the bacteria, our desirable gene is there. That gene undergo transcription and translation to produce more and more amount of protein. So you should keep that bacteria in where? A continuous culture system. So what is the right answer? If you wanted to get the protein, recombinant protein in high yielding amount, you should keep the transformant bacteria in a continuous culture system where they get the food, oxygen, and they multiply more and more and more. It is called what? Bioreactor. If you are able to recall, bioreactor, after that what? Downstream process, purification and also separation. You got my point? Yes, absolutely correct. Right. So what's the correct answer? A continuous culture system. Should be. Who is that? Nadia. Absolutely correct, beta. The continuous system, we will keep the bacteria. So they keep on divide, grow exponentially. NCRIT mentioned, you know, 1,000 to 100 to 1,000 liter of protein we are getting. 1,000 liter of recombinant protein we are getting, not 1 ml and 2 ml, my dear student. That's why NCERT mentioned it is exponential growth. So where you should keep? In the culture medium. So right answer is option C. Next question, sir.
Coming to the next question, all these organisms that is coming in NCERT, you have to learn which of the following microbe is used for ripening of Swiss cheese. You know there are different kinds of cheese in the market. The flavor, texture, aroma, all these will be varying according to the organism that you are using. Swiss cheese formation, the organism that is used is, let me check the comments. Yes, it is Propionibacterium shermani. Remember, that is an example of bacteria, whereas Roquefort cheese is actually formed using a fungus. That is Penicillium roqueforti. Even you can see there is Penicillium camber tea, that is for producing the camber tea cheese. Then streptomyces, that also you should remember. Streptomyces is actually used in order to form antibiotics like streptomycin. So all these examples you should learn. Roquefort cheese production is using a fungi. The fungus is Penicillium roqueforti, whereas Swiss cheese formation we are using Propionibacterium shermani, and whereas camber tea you can see Penicillium camber tea only is used. Then streptomyces, that is used in the production of an antibiotic streptomycin. Streptomyces griseus, that is a bacteria that is used in the production of antibiotic, life against life. And also remember, these antibiotics are example of secondary metabolites. It's not primary, it's secondary metabolite. Once produced in a cell, it doesn't have apparent function in a cell. It can be used for different function. Now coming to the next question, hope you got it.
Look at here, one more question. Students, look at here, this is one of important question. Significance of heat shock method in bacterial cell. Significance, this academic year there is a high chance, my dear students, you should remember there are 4 methods are there to transfer the gene into host cell. So this is what, tell me, heat shock method in bacterial transformation to facilitate is given. Binding of DNA with the cell wall. They're asking cell wall, binding a DNA with cell wall. Next, uptake of DNA through membrane transformation protein, transport protein, the given. Number 3, uptake of DNA through transformant pores in the bacterial cell wall. Tell me here, what exactly the reason this or reason of this heat and shock treatment? Tell me, if you are able to recall, DNA is hydrophilic in nature, the bacteria, the host is not going to take this DNA. But we have to force the DNA to take. We have to we have to make competent host to take the DNA. You got my point? So what, for this process, what we have to add? Bivalent cations like calcium ions to take up. So what is the correct answer of this question? Very good, very good. So answer is what? Tell me. Absolutely, Venkata. Very good, beta. Very good. So uptake of DNA through transist pores in a bacterial cell wall. My dear kids, this time I'm repeating one more time. You should prepare thoroughly about the biolistic gene gun method in where to transfer the DNA into plant cell. Micro injection method to where into a animal cell, and this what they call competent to host to making in where bacterial cell. These 4 methods. One is chemical method also there. Polyethyl glycol, chemical method also there to transfer gene into host cell. My dear kids, this academic year there is a high chance of from this 4, so all of you properly prepare and go. Yes, Mike, absolutely correct, beta. Nadia, correct. Next question. Look at the next question.
Production of beverages at industrial scale require growing microbes in very large containers known as. So in the industrial scale production of beverages and all, first you can see there is fermentation using the yeast, and it will be done in large vessels, that is the bioreactor or the fermenters. So the answer is fermenters, and the organism that you are going to use is Saccharomyces cerevisiae, followed by the distillation. So in case of wine and beer, the distillation is not required, but in case of higher brands like the whiskey, vodka, rum, etc., you have to use some distillation techniques also. So that's all.
Look at the next question, students. Who among the following was awarded the Nobel Prize for development of PCR technique? The given polymerase chain reaction, very important process for diagnostic method. Do you remember DNA amplification method? In the NCERT, what is given? Molecular diagnosis, early diagnosis of diseases is possible by 3 techniques is given. Can any one of you guess what are the 3 techniques? One is called PCR, one is called ELISA test, third one is called rDNA technology. PCR is essential to amplify the DNA. RT-PCR, RNA amplifying. The PCR is what? DNA amplifying. So remember, PCR is awarded by Carey and Mills. Carey and Mills was a scientist who given this entire PCR technique, denaturation, annealing and extension. So very good. So very good. So what's the correct answer? So correct answer is option C, Carey and Miller. Cohen and Boyd, do you remember, student? First rDNA constructed by Cohen and Boyer by ampicillin resistant gene, Salmonella typhimurium and E. coli bacteria. Cohen and Boyer in 19 what? Can anyone of you tell me? 1972. First time. This one is what? This two Cohen and Boyer develop what? rDNA. First artificial rDNA they prepared. Harigovind Khurana. Do you remember? This is in genetic code. We learn about the Harigovind Khurana. Yes, students? Yes. Absolutely.
First comment by
Absolutely.
Hridya.
Who is that? Hridya, right? Right. Very
Hridya
good.
first
Better.
comment.
Very good.
Correct.
Next question, sir.
Coming to the next question, you can expect a question from this one. So many options are there, what they are producing and all, you might have learned. Aspergillus niger. Aspergillus niger is associated with, we are using that microorganism for industrial scale production of citric acid. Whereas Clostridium butylicum, that is actually used in the industrial production of butyric acid. Whereas Acetobacter aceti, that is used in the production of acetic acid. Lactobacillus, that is used in the production of lactic acid. 2143. It is 2. Hope you got it. So learn all the examples and the organism used in the production correctly. So let's go to the next question. All have answered. Very good.
This is very important, beta. In microbes and human welfare chapter, you should remember different type of what they call organic acid bacteria and also fungi, microbes. Okay. Next question. An antibiotic resistant gene in a vector usually helps in the selection of. Remember, my dear students, we discuss about the selectable marker. Any vector, right? Tell me, vector must have 5 characteristic feature. I said in the theory class. What is that? First one, it must have ORI, origin of replication. Number 2, it should have ROP, repressor of protein. This protein is essential for the initiation of replication. Next, it should have selectable, what beta? Marker. Selectable marker should be there. Next, number 4, it should be small in size, right? It should be what? Tell me, small in size. Next, what beta? The last characteristic feature of vector is it should have recognition sites also. Any vector you take, PBR322, you take tiplasmid, you take BAC, YAC, tiplasmid, plasmid, cosmid, lambda phage, M13, RI plasmid, any plasmid you take, this characteristic feature they should have. But what they're mentioning, okay, fine. Sir, what would happen if selectable marker is not there? Plasmid don't have selectable marker. My dear, if plasmid don't have selectable marker, how can you differentiate this is a transformer and this is non-transformer? Yes or no? How can you differentiate? Bacteria will tell, sir, I took your desirable DNA, sir, I am the transformer. So tell me, how can you identify? You have to identify, you have to distinguish, and you have to eliminate what? Non-transformer. I don't need you, get out from here. I want what? Transformer. Because my desirable gene with them. So they will amplify, they will give protein, my desirable protein in bioreactor. You got my point? So tell me, the antibiotic resistant in selectable marker, 2 important things we discussed. One is called antibiotic resistant gene. One is called ampicillin resistant gene and tetracycline resistant gene. Second one is called LAC-Z gene, beta galactose. This is called what? II. What is II? Insertional inactivation method, beta galactosidase enzyme. So tell me here, competent bacterial cells, transformed bacterial cells, recombinant bacterial cells, none of this one. So here you peoples are thinking this one, between these two will be get confused. Tell me, any one of you, what's the correct answer? B or C? B or C? No, my dear. Cross check. Mahek, it is not B. It is given transformed bacterial cell. No, it's a recombinant, because inside the transform there are 2 type of transformers, beta. One is called recombinant transformer. One is called non-recombinant transformer also there. One is called recombinant transformer, one is called non-recombinant transformer. So you have to identify what? Recombinant transformer. So we will go with what? Recombinant bacterial cell, beta. So answer is option C. Remember all of you, selectable marker or selectable marker, ampicillin, tetracycline and LAC-Z gene. Thoroughly prepare and go this time. I'm telling you, sure question can come. Good, very good. Yes, good. Recombinant presence of chromosome. Yes, absolutely. That is called what, beta? That is a LAC-Z gene. That is a LAC-Z gene. Good. Very good. Good, but next question.
Coming to the next question. Dash are used in detergent formulation and helpful in removal of oily stains in the laundry. So large scale production of lipase is actually done in the bioreactors and all, and this has been utilized in the formation of detergents. So the answer is lipase. Then let me tell about pectinase. This enzyme also has been produced in a large scale for the clarification of bottled juices. So bottled juices, when they are making, first you can see a lot of foamy it will be. So for clarifying, removing the pectin and all, they will be using pectinase enzyme. So this also, when you're watching, please try to recollect, this is for clarification of bottled juices, whereas in detergent, the enzyme that they are using for removing the oily stain is by adding lipase enzyme.
Okay, sir, can you ask any one of you tell me what is the function of this one beta ligases?
Ligase. What is the
Any one of you
function?
tell me what is the function of this ligase enzyme or ligase enzyme? Any one of you? Something very important. This is one of the tool of RDNA technology, right? This is a tool of RDNA technology. How many of you got my point?
Got,
yes,
sir,
yes,
got.
yes.
Dia
Absolutely.
answered first.
Absolutely. RDNA technology to without ligase glue, it is a Fevicol beta. You cannot bind two DNA particles if there is no ligase enzyme, right? Next question.
Yeah, first.
Yes, who is that?
Molecular glue.
Molecular glue. Very
Someone
good.
is commenting. Okay, very good, very good.
Next question. Look at this question, my dear students. Something is, I think, wrong statement. I think so, right? Which of the given statement correct? Okay, sir. Correct in the context of visualizing DNA molecule separated by agarose electrophoresis is given. Correct statement. DNA can be seen in visible light. Is it visible light? Can you see the DNA in visible light? Next, DNA can be seen without staining in the visible light. Next, ethidium bromide stained DNA can be seen in visible light. Next, ethidium bromide stained DNA can be seen under the exposed UV light. So answer is what? Absolutely correct. Very good, my dear. Flip flop. Welcome back. Correct, my dear. So answer me. What kind of color? Tell me here. It is bright orange bands are formed in electrophoresis. Separation of DNA fragment based on their size. Smaller size particles are move away and bigger size particles are on the. So that means a slow movement, right? So this is the correct answer. What is the correct answer? Option D, ethidium bromide. My dear kids, for the NEET purpose, ethidium bromide, remember EBR, and also what? UV light. Gel electrophoresis, my dear, if you are preparing biotechnology chapter, my dear, please, RDNA technology steps after that, gel electrophoresis, thoroughly prepare line by line. I have a question. Where you will upload, where you will load the DNA particle, negative side or positively charged side? DNA. If you are able to recall what I given the hint beta, it is negative acidic in nature. We discussed right in the class DNA, deoxyribonucleic acid, but we convert N into negatively charged. Very good, acidic in nature. So answer is option D. Next question. Sir.
Okay. Coming to the next question, select the microbe which is the source of clot buster. So removing the clots in the arteries, vessels and all, we will be using an enzyme, you know which is the enzyme streptokinase, and it has been produced by a bacteria, that is Streptococcus. And remaining options also, let's see, already we have gone through it. Lactobacillus, that is actually producing lactic acid and it's a bacteria. Then Aspergillus niger, that belongs to Ascomycetes, it's a fungus that is used in the production of citric acid. Then Penicillium notatum, that is used in the production of antibiotics, that is penicillin. So penicillin is the antibiotic that has been isolated from Penicillium notatum. So hope you know this. Now let's move to the next question.
Right. Next, this one also, my dear, sir selected a very important question, especially in microbes in human welfare. Bioactive molecules. These are actually bioactive molecules. Cyclosporin A, what they call streptokinase enzyme, and this all statins are bioactive. Very important. Which of the following statement does not hold true for restriction enzyme? They are asking does not hold true for restriction enzyme. It recognizes a palindrome nucleotide sequence. I don't know, correct or not. Come on, any one of you. Dia, Reem, Meet, and Yatri, Magashri, Astral, Usna, Ridya. Find out the correct answer. Correct or not? This statement right or not? It will identify the palindromic sequence. Okay, fine, correct. Sabas, good, very good. Next, it is an endonuclease enzyme, right? Restriction endonuclease enzyme. RE is a type of what? Endonuclease enzyme. It is isolated from viruses. Hey, it is isolated from virus. Next, it can produce the same kind of sticky ends with different molecule they given. What about different ends in a different DNA molecule? Now tell me here, restriction endonuclease enzymes are actually defense mechanism of bacteria beta. If you able to recall the theory classes, from 240 different type of bacteria we identify. So far we have been isolated more than 900 restriction endonuclease enzyme. First restriction endonuclease enzyme, first isolated Hind D2. Do you remember? Hind D2. Hind D2. H. Haemophilus influenzae. Yes or no, my dear? Yes, absolutely. Venkata. Correct, but Reem. Very good, very good. So answer is what? We are not going to be isolate restriction endonuclease enzyme from where? Viruses. Actually, you know, in 1963, because of virus, the scientists that identify this enzyme beta. Because bacteria are protecting themselves from where? Viruses. For that purpose, they are producing restriction endonuclease. The name tells us restricting the growth of bacteriophages in the bacteria. Viruses growth, they are restricting. So, restriction endonuclease enzyme. So, answer is what? Not true is we are not isolating from where beta? Viruses. Remember, my dear, restriction endonuclease enzyme entire line by line prepare. Nomenclature and function, what they're going to break, what type of sticky end, what type of blunt end. Okay, thoroughly prepare and go. Next question, sir.
Coming to the next question, an immunosuppressive agent which is used in organ transplantation by the patients. You already know what it is. So please comment which organism is producing Cyclosporin A. Cyclosporin A is an immunosuppressive drug. Yesterday also Sandeep sir has discussed the same. That means during kidney transplantation or organ transplantation, in order to prevent the graft rejection, they are actually giving this drug to suppress the activity of the blood cells, especially the WBC. So it is Cyclosporin A. Which organism is producing that? All of them commented. First comment from Reem only, Trichoderma polysporum. One more question, it belongs to which group of fungus? Is it coming under Phycomyces, Ascomyces, or is it Deuteromyces or Basidio? Please comment, it belongs to which group? So we are expecting that also because you learned it in biological classification. Long back. You people will give answer from 12th grade, but when the 11th grade question comes better, everyone will become silent. Everyone will become silent. I don't know why. Trichoderma belongs to which one? Trichoderma belongs to.
Who is that?
Yes.
Wait,
Vidya, correct only.
Question mark, look at the question mark.
Is it correct?
Doubt, sir, so that's why question mark. Good, better.
Okay, good answer. Deuteromyces, it belongs to Deuteromyces.
Now, next, look at here. Which of the following columns and choose the correct option from the codes given below is given. Code given below is given. Ribonucleotide, chitin, cellulose. Tell me, come on. Cellulose is for what? Tell me. Cellulase. Chitin, chitinase. Ribonucleotide enzyme, ribonuclease. Sir, is this a NEET type of question? How simple question? That is a 2019 they asked beta in AMPT. 2019 a simple question they asked. So answer is 312. Where is 312? 312 option is B. Simple question. I am not going to waste time. Simple question. Next question, sir.
Next question. The product of Monascus purpureus has been commercialized as. You know what is the product of Monascus purpureus? Last question also the option had it, that is statins. So you know what is the function of statin? It's a blood cholesterol lowering agent. Then already you know about the immunosuppressive agent. Just now you answered Cyclosporin A, that is produced by Trichoderma, clotbuster, streptokinase produced by Streptococcus. Then clarifying agent of bottled juices, remember pectinase.
And moreover, students, 1 minute, sir. Here it is there, when you're reading this question, it is given, it worked like a enzyme Competitive inhibitor. Competitive inhibitor of cholesterol making enzyme. Beta, if you have NCERT, read and underline this one. This clot booster. What is the Monoscus purpura? Statins are actually behave like a competitive inhibitors of cholesterol making enzyme. So especially are selected very important question beta from bioactive molecule. This chapter is easy only, but you people will ignore. You people will not properly prepare. Yes or no, sir? This what they will tell. Okay, this
And please note 6 question in 2019 question paper of NEET exam. Refer it. 6 question from microbes.
Microbes.
So select the paper, go through it. You will find it.
Chapter
6 questions.
Don't ignore. Next question. Next question. The selectable. For selectable markers. For selectable markers, it helps to select the host cell which contain the vector and eliminate the non-transformants. Okay, genes encoding resistant to antibiotic like ampicillin, chloramphenicol, tetracycline, kenamycin are useful selectable marker for E. coli. Which of the following statements, which of the statements are given are correct? How many statements are correct? Any one of you. Come on. Sir, selectable marker, just now only I said, what the purpose of selectable marker? It helps in the elimination of non-transformer. It helps to select the host cell which contain the vector for the elimination and the elimination of the non-transformer. Statement correct. Beta, selectable markers are the one only, right? It is telling this is a selectable, this is the recombinant and this is a non-recombinant, this is a transformer, this is a non-transformer, right? So answer is what? Both statement. Hold a minute. Here it is given what? Sir, genes coding resistant antibiotic like ampicillin, chloramphenicol, tetracycline, kenamycin. But in PBr322, two of them are not there. What are the not there, beta? I think kenamycin is not there. Chloramphenicol is not there. Am I correct? Yes, beta. These two are not there. You know, normal E. coli vector does not have any one of these four. You know, normal E. coli bacteria plasmid is there, right? Does not have any one of these four. So remember, all of you, you will get a question PBr322. Which one? Where is the? Remember one more important thing. When you look at the PBr322, right side is ampicillin. Left side is what? Tetracycline. In the tetracycline, two recognition sites. Two recognize the BAM, H1 and SAL1. And here it is PVU and PST1. Remember beta, this question very important. Good, very good. Next answer is what option? Both statements are correct.
Coming to the next question. Treatment of wastewater is done by photoautotrophic microbes. So here photosynthetic microbes we are not using in the secondary treatment. You know that the wastewater treatment will be having primary and secondary treatment. Primary treatment, it include only sedimentation, filtration, as well as removing the floating debris and all. And the effluents will be given to the secondary treatment tank, where you can see the aerobic organism will be utilized first. That is naturally present inside the sewage, and it will not be including chemoautotroph. It will be including heterotrophic microbes that are naturally present in the sewage. Okay, all of them are commenting. First comment from Magasri. Magasri has commented first. So it is heterotrophic microbes that are naturally present in the sewage only, that has been used in the secondary treatment. And it's not the inoculated from outside and all. They are naturally present already. So that's all about the sewage treatment.
Next question. This is most important question, beta. You should pay attention to identify the answer. Look at the question. The question is telling which of the following statement is incorrect. They're asking what? Tell me incorrect statement. Come on, Diya, any one of you try. Venkata, try my dear. Try Mahek, try this one. What is given? Retrovirus have also been disarmed and are now used for desirable gene into animal cell. Retroviruses. Gene therapy, we use retroviruses. I told you the part of RNA which is pathogenic, remove. After that only it is possible to introduce in the animal cell. Downstream process is one of the step in rDNA technology. DNA is a positively charged molecule. The presence of chromogenic substrate give blue colonies. If the plasmid in the bacterial does not insert, how many statements are wrong? Come on. How many statements? Absolutely. Who is this, sir? Who is this such? Yes, good, very good. Venkata, correct. Diya, correct. Magashri, absolutely correct. Remember, third one is DNA is positively charged given beta. I told you it is negatively chargeable. This process correct. This one is correct. Third one wrong. What's wrong in that fourth one? What's wrong in the fourth one, my dear? The process of chromogenic substrate give blue colonies when our gene insert. When our gene insert beta-galactoside enzyme inactivated, no beta-galactoside enzyme come which cannot convert chromogenic substrate LAGZ into what they call normal substance. So tell me, it will be remain white in color, so colorless. So tell me, what's the correct answer? Yes, absolutely. The right answer is what? So this one is also wrong statement. So incorrect statement is 3 and 4. 3 and 4 is option D.
Next question. In sewage treatment plant, primary treatment is, already you know what is primary treatment. We already has discussed previously. So whenever we are taking the sewage, we will be doing the filtration, sedimentation and all. That will be the primary treatment. So here it's the physical process which involves sedimentation only is wrong. It is the physical process which involves both filtration and sedimentation. Biological process is secondary treatment. So biological process you can first of all cut it down. So it is the filtration and sedimentation. After that you can see the substance will be undergoing physically sediment and the top effluents will be taken for the secondary treatment. That is biological treatment.
Right, very good, very very happy. You all are giving nice answers. Diya, Reem and Search. And who is this? Ridya, absolutely correct. Venkata, you also doing very well. No problem. If it is wrong option also, no need to delete, my dear. It's keep it with you, no problem. I will correct you. Okay, this is the right time to realize. This is the right time to recover, revise all topics. Look at here, what is given? Identify the correct statements is given. Identify the correct statements. Okay, it is a big question, statement based question. Here after all statement based questions only, they are asking what correct statement is given here. Chalo, we'll see what is the correct statement is given. The first recombinant DNA was constructed by using piece of DNA from plasmid carrying antibiotic resistant gene in the bacterium Salmonella typhimurium linked to plasmid of E. coli. Statement 1 is right or wrong? Statement 1 is right or wrong? Tell me fast. Statement first one is correct statement. Okay. When cut by same restriction enzyme, the result DNA fragments have same kind of sticky and these can be joined together by DNA ligase. How simple. This is also correct. This is also correct. What's wrong in the third one? I'm not getting what's wrong in the third option. Now, the presence of more than one recombinant sites within the vector will generate several fragments which will be simplify the gene cloning. Oh my god, if you read the NCERT line by line and if you understand the concept beta, if you use more and more structured endonuclease enzyme, more fragments will form. It will make you complicate is given. It is given simplified is given. How many of you? Absolutely, very good, my dear. Absolute. If that's why I'm telling you, find out the find out the incorrect statement and practice more correct and incorrect statement. Statement based questions. Practice sample paper. Practice PYQ papers of NEET. Statement based questions are given beta. Statement based questions cover lot of portions. So this is not simplified. It is actually complicated. So correct answer or incorrect answer? Correct answers are 1 and 2. Where is 1 and 2? 1 and 2 option B. Sir, look at the correct option. How many are given?
So many.
B. Right. Good.
B.
Very good. Absolutely good beta. Next question.
Next question. During primary treatment, all solids that settle forms dash and the supernatant forms dash. Let me check what is the correct answer. because Venkat was commenting many times. Oh, now also he is looking at the highlighting, but that highlighting is wrong, Venkat. The answer is not 3. The answer is primary sludge. Got it? So understand, solids that settle down, it will be forming primary sludge. And the top one that will be coming, that will be forming the supernatant, which will be taken for the secondary treatment. So always don't think that is will be right. Sometimes it may go wrong. So read the question and the options carefully. Hope you got it, Venkat. Now let's go to the coming questions also.
Next question. This is also statement based question, my dear. Statement based question. You should peacefully think, you should understand what we learn, you should recall what we learn in the class, during the class time. Look at here. Read the statement about the gene gun method. Gene gun method is also called what? Biolistic method. To transfer the gene in where? Plant cells. Now look at here what is given. This method is also known as biolistic technique. Oh, I only given the answer, my dear. First question, first statement, you don't need to think. So this is the correct answer. Okay, fine. This is the method cells are bombarded with high velocity microparticles of gold, tungsten, coated with RNA plants. RNA in plants. Coated with RNA or DNA. Coated with RNA or DNA? I am not getting this. So any one of you help me out. It is what? Tell me. It is what? Tell me. It is DNA, not RNA. Read the question carefully, my dear. Take time. 3 hours time is there in examination hall. Here also, one question, 1 minute. Thoroughly read, properly read. So something is wrong there. They're asking what statement? Which of the statement given about correct statement? They're asking. Okay, fine. Important crop like maize, rice, wheat have been transferred by this method. These all are plants only, right? Krishna sir, these all are plants.
Yes.
So that means answer is what? Tell me. This statement is correct. This statement is correct. So final answer is what, beta? 193. Sabas. Very good. So first statement correct. Second, third statement correct. Second statement instead of what is instead of DNA? They given RNA. So answer is 193 statements are correct.
Don't change, sir. 1 minute. One person is telling it is highlighted.
Where?
So answers.
It's highlighting.
Okay, now let's see how they are going to find the answer.
Who is
now
that?
Venkat. Venkat only was commenting many times. Last answer was wrong also.
But what?
Okay, let's see. What are flocks actually made up of? Now, I'm expecting answer from Venkat. What will be the answer? You know what is flocks? In the secondary treatment or biological treatment ranks, you can see they will be forming a mesh-like structure that is made up of some bacteria and fungus, forming a filaments. So what is the answer? So many are commenting. Okay, this time the masses of which bacteria? Is it anaerobic? No. And is it aerobic fungi only? No. It's masses of anaerobic bacteria and fungus, of course not, in the primary tank. So you can see in the secondary treatment, the first tank we will be using aerobic bacteria that only will be forming meshes along with the fungus and all. So the answer is D. Hope you got it. Many has commented.
Venkata, are you there?
Yes, he's there now.
You got my point, but I don't worry. No problem.
Okay, let's
good. Next question. Good. Next question. Any one of you identify the incorrect statement? Any one of you help me out? Come on, fast. Which of the following statement incorrect? Which of the following statement incorrect? I will read, but I don't know. You have to find out the answer, my dear. Just I will read. Eco R1 cut the DNA between bases GA. They're asking what? Let me mark this one. Incorrect statement. Okay, fine. Making multiple identical copies of any template DNA is called cloning. PBr322 is a natural plasmid. Agrobacterium tumefacium Thai DNA is a natural genetic engineering. Something is incorrect is given. What is that? Something is incorrect. My God, who is this? Reem, Mahek, Venkata. Venkata is now lied. Now Venkata Krishna is an all. Diya, why question mark, my dear? Remove the question mark. PBr322, P plasmid, Bolivarov, Rodriguez. 2 scientists identify this, constructed this. So it is not a natural. What is the artificial? This is artificial one. So answer is what? Incorrect statement C. Eco R1 between G correct only. Eco R1 between G correct only. Anyone? Good. Next question, sir.
Sir, you change. Let me check. First
One
of
more
all,
time,
option we will
Okay.
change first. Okay, now let's see.
Fine, fine. Come on, come on.
Okay, now let's see who is going to answer first. So here, the ladybird beetle and dragonflies are useful to get rid of. This is coming under biocontrol, one expected question, because usually the example part and all everyone will be skipping. So here ladybird beetle are actually natural predators against aphids, and dragonflies we are going to use to get rid of mosquito. The answer is D. Hope you got it. Now let's start using. Okay, Reem answered the first time. Okay, we have seen it. Reem, very good.
Good,
Keep
good,
on
good.
answering like that.
Very good.
So we should use organism for biocontrol rather than using pesticide, chemicals, etc. So biocontrol, one example that is given here, you have to learn it nicely.
If Reem is physical class, you would have give answers like fast. Very good, beta. Very good. Hyperactive girl. Which of the following statement incorrect? Again, incorrect statement. Now, Reem, try beta. Diya, Ridya, come on, try this one. Incorrect statement they given. Which of the following statement is again incorrect? Statement based question. Statement based question. Come on, come on. Find out, find out, find out. Each restriction endonuclease recognize the specific palindrome sequence. Fine, correct. Specific base sequence is known as recognition sequence. Fine, correct. Restriction enzyme can cut only one strand of DNA. Correct. Restriction enzyme belongs to the enzymes called nuclease. Is this correct? The C is correct or D is correct? Something wrong is there. I said it's correct, but is this correct? C is correct. Come on, come on, come on. Yes, absolutely. Very good, my dear students. You are very, very, very absolutely correct. Here it is given. Restriction enzyme can cut only one strand of the. No, 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 no. Wait a moment. GAATTC CTTAAG 5 prime and 3 prime 3 and 5 prime. When you use that, what they call scissors, molecular scissors, it will cut phosphodiester bond, hydrogen bond, and separate the DNA molecule. That means it going to cut both DNA strand. And moreover, if you have a knowledge on restriction endonuclease enzyme, what we discussed, restriction endonuclease does not act on single stranded RNA or DNA. It only acts on the double stranded DNA. It cut both DNA strand. But they mentioned cut only one strand of DNA. No, 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 no. It is what tell me incorrect statement. Incorrect statement is option C. How many given, sir? Given almost?
All.
Very good, very good. Next question.
So
Playing like anything with student.
that is good, no? Now, next question. What is the composition of biogas? You know about biogas production. The bacteria that we are using in biogas production is dash. Can you please comment which organism is actually used in biogas production? And second, in the biogas plant we will be adding so many organic material, and in that we will be putting some bacteria also. Which organism? First answer by Dia, methanogens. Methanobacter only we are actually adding in the biogas plant. We will be putting some cow dung as a slurry with water, so they will be having this methanobacter. And this methanobacter belongs to which group in kingdom Monera? Are they belonging to Archaebacteria or are they belonging to Eubacteria? That also I want you to comment first.
For the 11th grade topic, khatam. Everyone will become silent. But let me try. Let's try it. Ridya, Astral, Naina, Ridya, try. Methanogen bacteria, sir, belongs to what?
Given, sir. Ridya given. Ridya is revising.
Remember.
Okay, now let me conclude the answer. The gases that are produced in the biogas plant already all have commented: methane, carbon dioxide, and hydrogen. That is the answer. Hope you got it. Now let's move to the next topic.
Now look at the next question. Next question is assertion and reason is given. Assertion and reason is given. This is assertion reason based question from type plasmid Agarobacterium tumefacium, which cause dicot plant cancer called crown gall disease. I said, right? The tumor induced plasmid, type plasmid of Agarobacterium tumefacium acts as a cloning vector in recombinant DNA technology. Absolutely correct. Assertion correct. Assertion correct. Good. Next. a Thai plasmid which is used in the mechanism of delivering gene to a cell remain pathogen. If it is remain pathogen, why would we use, my dear? We will, that's why NCERT what is given. Now, Agrobacterium tumefacium Thai plasmid has been modified to deliver the gene into the plant cells. No more pathogen is given NCERT. No more pathogen. Even retrovirus also in the gene therapy, when you are transferring the desirable gene, ADA gene into the lymphocyte, you will be inactive that one. Whatever the genes are causing HIV, whatever the genes are causing the disease, you should inactive that one. So this is wrong, this is correct. So I can go the if assertion A, true or false. Naina, Ashlil, Diya, Kavya is given beta. Mahek, very good, very good. Next
First
question,
by
sir.
Kavya. Shall we start? No. This already they will answer, sir.
Try beta. Start doing very full fun today. Chalo, come on, all of you answer.
Which fungal extract was extensively used in the treating wounded American soldiers in World War II? Already you may be knowing it. So it's extracted from which organism also you may be knowing, that is penicillin. Penicillin has been extracted from Penicillium notatum. And two people were getting Nobel Prize for the same. You may be knowing them, Florey and Chain. First it was discovered by Alexander Fleming, accidental discovery. He was growing some bacteria in the laboratory. Unfortunately, some fungal spores were falling on that and they were retarding or killing the microbes or bacteria. So from that only he understood this Penicillium notatum is releasing some metabolic waste that can kill or retard the growth of other organisms. So life against life, that is antibiotics, first discovered by Alexander Fleming. Later the chemical was extracted by Florey and Chain and it was used in treating the soldiers that was affected in World War II.
Next question, biotechnology, accession reason question. Foreign DNA and vector DNA cut with the help of ligase. Foreign DNA, vector DNA cut with the help of ligase. Ligase acts by the forming phosphodiester bond. We know, carefully look at beta, ligase enzyme function is what? If you cut the DNA fragment, the phosphodiester bond, the 5' 3' will be free, right? So ligase enzyme function is to join this one, making a bond. In biomolecule chapter 11th grade, different type of enzyme classification is there. Ligases enzymes are the one which make bond, which helps in the creating of phosphodiester bond. So what is given? This is absolutely correct. I can go with this one. But what's wrong in the first accession? Anyone of you tell me? Accession is wrong because, sir, hold a minute. DNA, vector DNA cut with what? Restriction endonuclease enzyme. What? You given ligase enzyme here. How come it is? Yes or no? So I can go, this is wrong, this is right. Is there option is there? Yes, option is option D. Sir, cross check, option D is there. Kavya, okay. Naina, are you there? Astral, Vidya? Right, good, very good. Next question, sir.
So here, the greater BOD of wastewater relates to. So in the secondary treatment, you should understand when the biological treatment is done, they are trying to decrease the BOD of water. When the BOD of water is increasing, you should understand the pollution is high. So the main objective of sewage treatment is for treating the water and to decrease the biological oxygen on demand or biochemical oxygen on demand. So when the BOD is decreasing, the pollution level is less. When the BOD is high, understand so much oxygen has been used by the microbes to break the organic material. So dissolved oxygen content of the water will decrease. So BOD when increases, understand dissolved oxygen is decreasing and the pollution is very high. So when BOD is decreasing, understand dissolved oxygen is high and the pollution is less. So the greater the BOD of wastewater, understand there is decreased oxygen content of water and the level of pollution in the water. So greater the BOD, less is the dissolved oxygen and the pollution is very, very high. This you should correlate and learn.
Right?
Okay, very good.
Shall we
so
start?
many
We'll go very fast, better. Look at here. Next question, 35th question. Again, accession reason. Use of chitinase enzyme is essential or necessary to isolate DNA from fungal cell. Flip-flop. Last year also we had a one more flip-flop, flip-flop beta. He used to give super answers. So now today also you're giving very nice answer. I don't know both are same or not. But last year we had one flip-flop comment. That means one person. Very good, very good. Now is it correct statement or not? Reem, is this correct statement or not? Venkata, any one of you tell me, sir, it's true, absolutely. You need to isolate DNA from fungal cell by the adding of chitinase enzyme. So this statement is correct. Fungal cell wall made up of chitin. Chitinase is able to digest is how simple one. So what is the correct answer? It will be B or A. A or B? Come on, answer me. A or B? Who is the first one, sir? Who is the first one? Good. Reem, absolutely correct. Very good, beta. Very good. So correct answer is what? It is a right explanation. Right answer. Very good. Very good. Very good. Next question. Who is that? Next question.
We have to change it. So let's see. Okay, all are correct with respect to BOD, biochemical oxygen demand, except. So it refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. So they are actually correct only. So how much oxygen is utilized by the bacteria in 1 liter of water? That is actually the BOD. Then the BOD test is measure of the organic matter present in the water. So more the BOD, more is the organic matter and more is the consumption by the bacteria. Then greater the BOD of wastewater, less is the polluting potential. Greater the BOD means it's not less, it's actually high is the polluting potential. So the answer is 3. Hope you got it.
Right, look at the next question. NEET 2017 PYQ question. Look at the question. The process of separation and purification of expressed protein before marketing is called. What is the correct answer of this question? The question. Process is called. What is the process? Separation and purification. Expression in bioreactor. What is separation and purification, preservating, and also what? Quality control. That's what is exactly the process. Come on, fast. Very good, very good. One is called upstream process, downstream process, bio process, post-producing process is given. It is what? Downstream process. Remember, right? So, right answer is option B. Next one.
Let's move. Okay. Now, the Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan. So what is the purpose of this? To save these major rivers of our country from pollution, it is proposed to build a large number of sewage treatment plants. Under these plants, only treated sewage may be discharged into the river. So actually Ganga Action Plan and Yamuna Action Plan, when you are reading that concept, don't think it's associated with these two only. It's associated with all. The main purpose is to decrease the pollution level in most of the water bodies in the country. So all the statement given here is actually correct. Next question.
Next question. Can any one of you find out the right answer of this question? It is given here. Identify A, B, C. Bioreactor is given. A, B, C, D and E is given here. It is a simple styrate tank of bioreactor. Find out. Correct answer. Come on, find out. Yatri, try beta. Try what exactly the correct answer. Venkata, Yatri, Diya. Look at this one. Bioreactor is given. A is, here it is, this one is actually motor, I think so, right? A is motor, right? Very good. Who is that one, sir? Who given A? Who is that? Is that Zainaba? What is that? What's the name?
Harshan actually given first.
Good, very good, also. Absolutely. A, motor. B,
actually
foam breaker. This is foam breaker. C, sterile air. D is what? Tell me. Stream for what? Sterilization. And E is pH control, acidic pH control. So simple answer. Vidya, the right answer is option A is the right answer. Next question.
Sir, Mukesh Ambani is attending our class.
Mukesh Ambani attending. Very good, beta.
Now next one. Secondary treatment of sewage. Removes grit and large pieces of organic matter. So secondary treatment, is it removing the grit and all? That is associated with the primary treatment. Then it involves shredding, churning, filtration and sedimentation. That is also coming in the primary treatment. Does not require aeration. Actually, in the secondary treatment, first the effluence is passed to an aeration tank where the flocs are actually formed. And in that you can see they will be moved to the next tank where anaerobic treatment will be done. Then it involves microbial digestion of organic matter. So it will be grown both in aerobic and anaerobic tanks. Then activated sludge also will be formed, which will be sent to a sludge digester. And some of them will be sent back into the first tank in the secondary treatment also. So here the answer will be involves microbial digestion of organic matter. Hope you got it.
Mukesh Ambani sir, give me answer please. Though you're busy. Good. Is you giving? Right, next question. Next question. Look at the next question. Primers are. Can any one of you answer this one? Primers. What are primers? In DNA replication we have done. We discussed. In PCR we have discussed primers. Any one of you? Fast. Come on. Primers are small chemically synthesized oligonucleotide, about 10 to 18 nucleotides that are complementary to the region of template DNA. Chemically synthesized oligonucleotide, about 10 to 18 nucleotides that are not complementary. Double-stranded DNA, leave it. Double-stranded DNA, specific sequence present in the recombinant DNA, leave it. A and B. If you able to recall in DNA replication, tell me beta here, this is in PCR also, this is 3' and 5' and it is 5' and 3'. Tell me primers always add. Tell me any one of you, primers we will add opposite of which prime of parent? Tell me. Primers always opposite of 3'. So what will be this one? 5' to 3'. Here also what? Tell me, here it is 5' and 3'. So tell me, new DNA will be formed. New DNA will be formed from 5' toward the 3'. 5' toward the 3'. How many times I said new DNA always polymerization takes place from 5' to 3'. But here this structures are actually primers. Beta, primers are a 10 to 18 nucleotide complementary. What does it tell me? Complement. So answer is option A.
Now next question. What is the percentage of methane in biogas plant? Can you comment? What is the percentage of methane in biogas plant?
If you know the composition of methane, that biogas, you come to know beta. What is the percentage?
What is the percentage of methane in biogas plant? Let's see who is going to comment.
Fast.
Any comments, sir?
What percentage?
Any comments?
What percentage? 30, 40, 50, 60, 65. What percentage? Who is this Venkata? You're deleting messages more than your answers, my dear.
He's waiting for the answer there, sir.
Now tell me.
50 to 70%. So out of the given option, it is 50 to 70%.
Mahek and Reem, good. Ridya.
Okay, now only answer came from Venkata. Okay, Venkata, at least once you tried.
Good. In class also it's like that. Good. Next question. Look at the next question. Selectable marker is used to. Simple question. We have already done this one in the previous questions. Come on, any one of you. Selectable markers help in eliminating the non-transformers so that transformer can be regenerate. Identify gene for the desirable trait. No, identify gene trait. We are not going to be used the selectable marker. Select suitable vector. Transformation is a specific crop. Select suitable vector for transforming specific crop is what? Kuch lena dena hi nahi beta. No relation to this one. Leave it. Mark a gene of a chromosome isolating using restriction enzyme. Leave it. So the right answer is what? A is the right answer. Next question, sir.
Coming to next question. In biogas plant, for biogas production, the first organism that is carrying out the necessary things is which organism? Is it methanogen? Is it decomposer? Or is it a parasite or aerobic bacteria? 4 options you have. So in methane production in the biogas plant, which is the first organism that is acting upon? Is it a parasite or is it a aerobic bacteria? Is it decomposer or is it the methanogen? So please make a comment. Let's see.
They're not getting it.
So which organism is first involved in the biogas? Diya answered. So at least one got the answer.
With question
Decomposer.
mark. With question mark. Look at
So-
the question mark is given.
Diya, very good.
Good, better.
It's not methanogen. First step in biogas production is carried out with the help of decomposers. After the decomposers break down the organic molecules and all, some acid is produced. From that acid, the methane is released by methanogen or the methanobacter. So understand, first acting organism is decomposers. Venkat, why deleting the message?
okay.
At least now you answer.
Next question, 45th question, beta. 5 more questions, very fast. We are going to finish. Now look at it. This is the 2019 question, PBR 322 question. 2 antibiotic resistance genes in vector PBR 322. How simple? How simple? I don't want to waste time. You know everyone. Ampicillin and chloramphenicol. No. Good, Sabas. Very good. What's the right answer? Ampicillin and tetracycline. So answer is option A.
Now next question. Beside paddy field, cyanobacteria also found inside the vegetative part of. This question is actually associated with your grade 11 topic. That is the plant kingdom as well as biological classification. Let's see. Anyway, comments are coming. Cycus. Cycus is the answer. Now, my question is not that. Actually, in Cycus, which cyanobacteria is found? You might have learned one term in plant kingdom chapter, that is coralloid root. So, what is coralloid root of Cycus? Try to recollect. In plants like Cycus, around that you can see there is an association of a blue-green algae, that is cyanobacteria. So, this is actually called coralloid root. And the organism that is, it's not nostoc, it is Anabaena cycadae. That is the organism which is found in the coralloid root of Cycus. Hope you got it. Then Pinus, you might have learned about mycorrhizae, that is symbiotic association.
Yes,
in that you can see there is growth of fungus around the root of Pinus, that is mycorrhiza, whereas coralloid root is seen in Cycus, where you can see the nitrate content, they will increase and the fertility will be increased.
Gymnosperms we learned 11th grade, long back. Majority of students forgot, I think so.
Again message deleted.
Right, next question, 47th question. Identify what they call A, B, C, D in the given E. coli bacteria. A, B, C, D given diagram. E. coli, E. coli cloning vector. PBR32 vector. A, and here it is B, C and D is given. I told you, if you stand like this beta, this side is right side, this side is left side. Right side is, I told you, ampicillin, left side is tetracycline. So I'm writing it is tetracycline. Here it is BAM H1. Am I clear? BAM H1. Next here it is tell me C after ROP ORI. D is what tell me ampicillin resistant. If I am not wrong, this is ECO R1. A is ECO R1. Any one of you given correct answer? Good, very good. So ECO R1, BAM H1, ORI and also ampicillin. One more important thing, PBR322, 8 recognition sites. Okay, beta. 1 ORI, 1 ROP. Next one is called 2 selectable markers. Remember this question, PBR 322 this academic year, high chance is there, my dear kids. All of you, PBR thoroughly prepare and go. All of you. But we will use the restriction 1, 2, 3, 4 only we will use beta, because in the particular area only what is there? Selectable markers are there. So answer is option A. 48th question.
A nitrogen fixing microbe associated with Azolla in rice field. In rice field, we will be putting a fern, Azolla fern, and it increase the nitrate content because its leaves are actually associated with a cyanobacteria, and the name of the cyanobacteria is Anabaena. So anabina has symbiotic nitrogen fixation. It's associated symbiotically with the root of cycas, as well as you can see it's symbiotically associated in the leaf of azolla fern. That is why azolla ferns are actually added in the paddy field and rice fields. Hope you got it. So remember the name also, Anabina azolla, that is the species which is found in the leaf of azolla.
Last question from biotechnology principle and process. This is the last question, but the next question is again microbes and human welfare. Today we are winding up this session. Look at here, 1, 2, 3, 4 questions are given. 4 questions and simple question. How many restriction enzymes are isolated till now? How many? 900. Very important. Good, very good. Number of bacteria strain which are restriction enzymes have been isolated, 230. In the last question beta I given 240, just correct it. So subject corrections, 230, from 230 900 different type of, more than 900 different type of restriction endonuclease enzyme. But what about this question and this question? Last 2 questions. Carefully answer this one. Look at here, what is given. How many fragments will be generated if a linear DNA molecule is digested with a restriction enzyme having 4 recognition sites? Okay, linear DNA, like this linear DNA, 1, 2, 3, 4 places I cut. 4 restriction endonuclease enzyme I use. So tell me, this is 1 fragment, 2nd fragment, 3rd fragment, 4th fragment, 5th fragment. So what's the right answer? Absolutely correct. Absolutely. If you read the question beta, if it is linear DNA, number of restriction endonuclease enzyme plus 1 you should add. Answer is 5. You got my point? You got my point? Come to the last question. Last question. How many fragments will be generated if closed circular DNA molecule is digested with the restriction enzyme having 6 recognition sites? Let's take 6 recognition. 1, 2, 3, 4, 5, 6. Fragment 1, fragment 2, fragment 3, 4, 5, 6. So number of restriction endonuclease enzyme equal to number of fragments in where? Circular DNA. So answer is option B. Am I correct? Am I clear? Very good beta. This type of question you have to prepare and go 2 mark or maybe case study or maybe direct NEET question they can give. Am I clear? So this is last question of biotechnology. The next questions are today's last question.
Done, sir. Yeast is used in the production of, you already know, Saccharomyces cerevisiae that belongs to Ascomycetes family, the unicellular fungus, and it has been used in the production of. You can make the comment. You already know what is the answer. So we are expecting the comment. Already we are winding up the session with that comment. That is bread and beer making. We are using that yeast. So that's all about
today's session, my dear. I hope you enjoy these 2 chapters, microbes and human welfare and biotechnology. And this session highly help you to get 100% good marks in your upcoming NEET exam and also theory exam. Yes or no, sir?
Yes. Because we were touching most of the topics. The questions were taken in that type only, the arrangement also, touching most of the concepts in both the chapters and all. So this might be very, very useful from the board exam point of view as well as coming in the NEET exam. So the weightage wise only the questions were selected and the repeated areas also. Hope you got a good idea regarding the revision.
Right, and moreover, remember all of you, tomorrow we are going to come with the 2 more chapters, biotechnology application, and 1 more chapter, organism and population and ecosystem we will select. Don't miss this session, student. This will be 100% help you to get good marks. And thank you so much for attending this session.
Okay, thank you all, because all were very cooperative and the comments were continuously coming and we were listening very nicely also, and we appreciate your cooperation also in attending this session, and all the best to all of you. We will be coming with more classes again.
Thank you. Good night all of you. Bye-bye.
Hi everyone. Welcome to
Amazing channel of Brilliant Katha.
When you're rubbing one body with another body, electrons are transferred from one object to another object. Okay, this process, this type of electricity is been given a name.
connected carbon sp3, then it's connected to benzene. See all of you, halogen connected. What is this carbon hybridization? Full single bond sp3, see then a benzene ring.
Denominator poles shouldn't be included in the denominator poles. That means the denominator, these -1 and -5 should never be included in the solution.
Homocircle caudal fin. It's giving 2 equal lobes. But in case of cartilage fishes, look at here, one big lobe is there, one small lobe is there. You cannot cut into 2 equal half this fin.